Hello, this is Thomas Warwick welcoming you to this bite-sized bio webinar, which today is sponsored by VectorBuilder. Today's presentation is titled How to Create Effective Vectors for Gene Delivery and is being presented by Connie Rich from VectorBuilder. Connie Rich is a former molecular biology researcher and instructional professor who joined VectorBuilder to bring gene delivery solutions and resources to other researchers and industry partners. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the top right of your screen, and I'll put them to Connie at the end. Note, we'll likely have a lot of questions today, so if you could submit your questions as they come to you, that would help us a lot. We have a few polls coming up during this webinar, so watch out for those, and there are also some useful handouts available to you as well, and they are available in the chat box on the right of your screen. Details of how to access the on-demand recording of this webinar will be sent to you by email shortly. So now over to you, Connie, for the presentation.
All right, thank you for the introduction. Hi, everyone, welcome, and thank you for joining us to talk all things vectors as we discuss how to create effective vectors, both choosing and designing them for gene delivery. Now today, I'm looking at this as kind of more of a gentle dive into vectors and gene delivery. I know many of you here haven't designed a vector in the past, so I want to make sure that we're all on the same page before we get into selecting and designing different vector systems. And that will include a lot of tips and tricks that we've learned at VectorBuilder over the years. Finally, we'll talk about how VectorBuilder can help you streamline your gene delivery research. But by the end of this webinar, my goal is to have everyone walk away with some foundational knowledge about vectors and how they're used in different gene delivery systems, as well as hopefully some inspiration to look at different systems and different approaches to try to find something that's really well suited for your lab in your research question. So let's start off with some kind of broad perspectives, some definitions, because gene delivery is a pretty big term and it encompasses a wide range of different applications and different approaches. But at its base, gene delivery is pretty straightforward. Really, all we're talking about is taking a foreign piece of genetic material and delivering it to your host cells. Now, this is pretty imperative for research in the life sciences as well as clinical advancement, but it's also very influential in a wide array of other industries as well. Now, for today, we're really going to focus on how gene delivery in the life sciences allows us to perform a wide range of studies, from observing endogenous gene expression to modulating pathways to editing a host organism's genome. The options for different experimental approaches is pretty vast. And the great and often daunting thing is that not only do we have a wide range of options for what we can do with that genetic material once it gets into the cells, we also have a wide array of options for how to get that genetic material into the cells in the first place. Now, this is where we bring in vectors. And vectors are essentially just the mode of transport for how to get your DNA or RNA into your host cell. Vectors take a variety of different forms, including viruses, plasmids, and RNA. And even within those options, there are multiple different subtypes as well. So if you start thinking about, we have plasmids and overexpression and lentivirus and shRNA and RNA and CRISPR, it can get really overwhelming. So what we're going to do today is we are going to just go through step by step and cover some key considerations as well as some tips along the way. But regardless of what application you use or what delivery system you use, luckily it all really starts the exact same way. It all starts with a plasmid. And probably you have encountered plasmids in one context or another, but we do often overlook the basic components of a plasmid, especially if you're just using one that's been in your lab forever or you've ordered one and you're using it as is. So let's take a minute to cover some of the really important components that we find on plasmids, and we're going to do that by dissecting the plasmid into its kind of life cycles, starting with its most basic function, which is replication in bacteria. Now, in order to do this, we're going to need an origin of replication, or ORI, which, exactly like it sounds, is our point of replication initiation. PUC is a really common one, especially for high copy plasmids. Additionally, we're going to want an antibiotic resistance gene. Ampicillin resistance is very popular, and this is going to allow us to select for and grow only the bacteria that have taken up our plasmid. This is going to allow us to create lots and lots of copies of the genetic material that we want to deliver to our cells. Now, this is often going to take the form of a gene where we have this open reading frame encoding a protein coding gene, or this can be an RNA, just something that we want to express. And once we have a lot of copies of this, then we can start thinking about how to get it into our host cells. But there are a couple of components that we still need to make this work and to make this identifiable in our host cells. We want to know which host cells have actually taken up this plasmid or other vector. So we're going to want a marker here that can either be another antibiotic selection marker, for instance, pyrimycin, or it can be a fluorescent screening marker like EGFP that allows us to actually visualize which cells have this vector. But in order to actually make this thing right here that we have worked so hard to deliver function, we do need a couple of other host-related components, these things that function in the host. So in order to actually express our ORF or our RNA, we're going to need a promoter that can drive expression. And if this is a protein coding gene, this ORF, we're going to also need a polyA signal typically that's going to serve as the transcriptional termination site, as well as addition of that polyA tail. And you're typically going to find this Kozak sequence here as well that's going to facilitate translation initiation. So now we have our plasmid. It's ready to go. If we can get this plasmid right here into a cell, it will function. Yay. But I mentioned before that there are a variety of modes of transport for our little gene of interest or RNA of interest right here, including viral vectors. So if you want to create a recombinant virus for gene delivery, there are some extra components as well as extra plasmids that you have to consider. Now, the plasmid that's carrying your gene or your RNA to be delivered is called the transfer vector, which is going to contain those host components and those bacterial components, as well as parts of the viral genome that are needed for delivery and or for increased efficiency. Additionally, we're going to have these packaging vectors over here that are going to carry other components that are needed to create a live virus containing your desired genetic material. Now, of note, these components are often separated over multiple vectors, and that can help to increase safety and ensure there's tight control over that viral production. So this right here is obviously a bit more complex than the regular plasmid we were just looking at, but the increased work on the front end can be really worth it. Now, while plasmids like this one right here are more simplistic in design and production usually, they and other non-viral vectors are just nucleic acids, so it's very difficult to get them across the cell membrane into our host cells. Instead, we rely on various different forms of transfection, and this can include things like electroporation. On the other hand, viral vectors do require a little bit more work on the front end, for instance, taking all of these plasmids right here and transfecting them into our packaging cells. However, this is a really well-established process, and it can be pretty straightforward to create this nice, easy recombinant virus that can then be transduced into either host cells or, entire organisms. And once they reach those target cells, these recombinant viruses can do what viruses do best. They infect cells. But instead of infecting with this pathogenic virus, it's going to deliver your gene or RNA of interest instead. So after we consider these sort of viral and non-viral options, we still have a number of decisions within those systems. So let's take a minute to discuss how we can choose not only between viral and non-viral systems, but also choosing within those options to get the right system for our model, for our application, and for our research question. We're going to start with a pretty high-level question, which is one that you should try to determine really early on. Do we want to use a non-viral vector, or do we want to use a viral vector? Both have a number of different popular options that we're going to explore shortly, but for non-viral vectors, all of these have to be transfected into those host cells. And this can be done using methods like electroporation, microinjection, or lipid nanoparticle delivery. Conversely, transduction, just you kind of expose the cells to your recombinant virus. This can often be a little bit harsher and or more technically difficult than transduction, especially if you don't already have this thing developed in your lab. Now, while non-viral vectors can be a little bit more work on the kind of delivery end, they are much easier to produce and are often more highly tolerated by cells, especially if you're putting this in a whole organism and doing in vivo transfection. You tend to have lower immune reactions. But in vivo delivery can be a little bit more difficult, especially if you're trying to target very specific cells, and especially if you're really developing this thing from scratch. But one of the biggest disadvantages for our non-viral vectors is that they generally have lower efficiency. Conversely, viral vectors have very highly specific and efficient delivery for particular viruses in particular settings, of course, which we'll talk about. But they do tend to have a higher immune reaction, of course, because they're viruses, and they tend to have this toxicity and can exhibit longer-term off-target effects depending on how they are designed and how they're used. Now, in general, with very, very broad strokes, non-viral vectors are a popular option for easy to transfect cells, particularly those in vitro, like hex cell lines, while viral vectors are really good for in vivo or for difficult to transfect cells. So let's take a closer look at some popular non-viral options. Now, the major differentiator between these three is the speed and length of effect. RNA is already transcribed, and so you're going to have very fast and very efficient activity. However, it does have lower stability, and host cells don't tend to like having just random RNA hanging around, so it gets degraded quickly and therefore has much shorter-lived activity. In contrast, regular plasmids do have to go through the transcription and or translation process, so they're going to be much slower to initiate activity. And they are more simple and more stable, which means that they're going to have longer-lived activity. And additionally, they can generally carry more cargo than RNA. Now, of note, it is less efficient in many cell types, including non-dividing cells, which RNA tends to be a little bit better at targeting. And you do have a risk of genomic insertion, although it is low for regular plasmid. Now, if genomic insertion is actually what you're looking for, then transposons are a really good option. They are actually designed for genomic integration, and there are a variety of options available, including Piggyback, Toll2, and Sleeping Beauty. Now, the downside here is that you typically have multiple vectors required, and because these are plasmids, they are going to have slightly lower efficiency, and they are going to be slow to have their sort of onset of action. So while you should keep in mind these advantages and disadvantages if you're considering non-viral vectors, typically it comes down to how fast and how long you want that activity. And actually, the same considerations are really important for viral vectors. Among popular viral vectors, we're going to really primarily divide these into viruses that stably integrate the gene or RNA of interest into your genome versus those that don't, those that have more transient expression. So for viruses that do not integrate into the genome, adeno-associated virus and adenovirus are our most popular options. And when deciding between these two, it largely comes down to how big your host components are. This is things like your promoter, your ORF, your marker. If they're less than 4.7 kb, we tend to go with AAV, whereas if they're larger, we go adenovirus. But a couple of important considerations for AAV is that it does have the benefit of very low immunogenicity if you're using this in vivo. And you can also take advantage of various different serotypes. So the capsid for the AAV virus, you can choose between different types of proteins that are present on that capsid that's going to give you either broad or very specific targeting or tropism. Now for our stably integrating viruses, lentivirus tends to be the go-to. It has a larger carrying capacity, and it also has the benefit of very broad tropism, being able to infect both dividing and non-dividing cells. Additionally, you can customize your promoter. Now, MMLV and MSCV, these tend to be more kind of historical viruses. A lot of people use them because they've always used them. But you do still see a good bit of use for cells like hematopoietic stem cells, which aren't very well targeted by lentivirus. And although those are our popular options, we do have some options for viral vectors that are less popular, but that are great for very particular applications. For instance, vesicular stomatitis virus, or VSV, can be really easily pseudotyped. This means that we can put different proteins on the viral envelope, allowing it to either have specific targeting abilities, or we can mimic other viruses and study viral entry mechanisms. Additionally, VSV as well as vaccinia virus are great options for vaccine development, partly because they have really broad tropism or targeting. And VACV, as well as herpes simplex virus, or HSV, are great candidates for oncolytic viruses. We actually recently posted a blog post solely devoted to how we can design VACV and HSV oncolytic viruses. And finally, rabies is really second to none when it comes to labeling neural networks. This is an excellent option if you are trying to study synapses, neural networks. Rabies is just your number one go-to. And as we're kind of considering which viral or non-viral option that we should kind of move forward with, we also have to consider some other key questions, including which application. Are we using overexpression, or are we using knockdown or knockout? Are we using mouse or human or zebrafish or Drosophila or plants? And how are we approaching those cells? All of these questions are going to help lead us to the right starting vector for our gene delivery research. But this is a lot of questions, so I felt like it was a good opportunity to go through a couple of examples. For instance, if we want to study a genetic disorder in mouse, for instance, a mutation that causes a gene to be inactivated, then we're going to want to use a CRISPR knockout. Now, if this gene that we're interested in is really, really important for early development, if we cannot make, for instance, this mouse without this gene being active, then we're going to want to target the adult cells. So we're going to need to deliver those CRISPR components to the adult using either a virus or possibly a plasmid or RNA. Now, as I mentioned before, this can be more difficult if you don't already have this set up in your lab. Another option is an inducible system where we deliver those CRISPR components, but we don't activate them until later on. Now, if this gene that we're targeting is not as important for early development, for instance, if we want to model muscular dystrophy, then we can micro-inject a plasmid or RNA at the one-cell stage. Now, of note, we do want to avoid delivery systems that involve that Cas endonuclease getting integrated into the genome and then expressed throughout the life of the animal, because that can lead to a lot of off-target effects. So for this, if we want to deliver these CRISPR components in the adult, AAV is going to be typically the better choice. And if we want to target that one cell stage, we can use mRNA for, again, that really short-lived activity. We make our edit, gets degraded, and we're good to go. Additionally, we could use lentivirus or transposon as long as we're using that with an inducible system. Now for another example, if we want to analyze a pathway that's containing a kinase, for instance, in a particular cell type like endothelial cells, then if we're targeting a very specific cell type, an in vitro assay is going to be really useful for us. And one of the ways that we can study this pathway is through overexpression. Now, because we're using differentiated cells in vitro, viral delivery is going to be a really good option because it's more highly efficient. And if we want to do a longer term study, then genomic integration is going to be our friend here. So considering all of those options, we're going to use lentivirus overexpression in this example. Now, I do want to emphasize that although these are good options for these particular examples, there is no perfect option and there is no single pathway to success where it's the only way that you can possibly have a successful gene delivery experiment. There are multiple options, but there are so many options that making informed choices is key to ensuring that you have good chances of success and your vectors are going to do exactly what you want. All right, we have our vector, we know what we need to do, and we have our little packaging systems all set up in place, or we have our little electroporation station, but now we need to put something in that vector, right? Now, whether you're using transposons or lentivirus, you're typically going to start with a preformed backbone. That's going to be the easiest option. That backbone is going to contain all of the necessary kind of almost background components. So for instance, in this regular plasmid backbone right here, we're going to already have those bacterial components in place. And for a virus, you're going to have the transfer viral components already in place as well. And you can optimize those, of course, but for today, we're really just going to focus on the sort of host components, the payload, if you will. And what we're going to do is take a step-by-step guide through each of these components and talk about key design considerations, especially related to the vector system that you have chosen. So we're going to start with the promoter that is actually driving expression of the thing you want. And there are a number of considerations that we have to take into account, but let's start kind of at the top with just what we are expressing. If we're expressing RNA, we have a few different options. So if we're trying to get RNA expression outside of cells for in vitro transcription, this is helpful if we want to create an mRNA that we then want to deliver to our cells, or if we want to create an in situ hybridization probe. Then in either of these cases, or in any case for in vitro transcription, a T7 promoter with T7 polymerase is going to be your go-to option. Now, if you want to do something in your cells, you deliver this vector to your cells and you want to create a small RNA, for instance, an shRNA or a gRNA, you have a couple of different options, but the most popular, the kind of standard, is a Pol3 promoter. This is a promoter that's associated with production of small RNAs. The most common is going to be U6. And for instance, if we're looking at different shRNA options, we have different systems to choose from, but U6 tends to be the most popular. U6 is going to ubiquitously drive expression of this shRNA, but we can only have one and we can only have it ubiquitous, expressed everywhere. The biggest benefit here is that you do have really high efficiency knockdown for those shRNAs. Now, alternatively, you do have another shRNA system that uses a different promoter. It uses a Pol2 promoter that's associated with production of mRNA leading to protein, of course. So for this other system called the miR30 system, you can have multiple shRNAs. You can also customize your promoter to be any different type of Pol2 promoter. The big downside is that you do have reduced efficiency of that knockdown. Now, finally, if you want to produce mRNA in cells, if you want to create a protein, you're just going to use a Pol2 promoter. But now we have a lot of other considerations that we have to take into account. So if we want to express a transgene, we have to now think about where, when, and in which vector. For the where, if we want it everywhere, we have a variety of different ubiquitous promoters to choose from. Now, each of these are going to have different strengths. What strength you want is a good kind of first question to ask. And it can seem obvious to say, well, I want the strongest promoter, but that's not always the best. If you have a protein that's going to be really efficiently translated and can potentially overwhelm the cell, or if you have a toxic gene, then a strong promoter is not going to be your friend. You're going to want a weaker promoter. Additionally, you have to consider which cells you are going to be targeting with this ubiquitous promoter, especially if you're trying to target every cell in the entire body, for instance, of your little mouse model. If you're doing an in vivo delivery and you want to target everywhere, you're going to want to avoid something like CMV, because CMV does tend to be silenced in some cell types, for instance, embryonic stem cells. So another ubiquitous promoter is probably going to be a better choice there. Finally, there are some considerations that are particular to lentivirus. We find in this lentivirus assay of various different ubiquitous promoters here that CAG does tend to be associated with lower titer and lower efficiency, presumably because of a high GC content. And additionally, you're going to want to avoid using the same promoter twice. For instance, the promoter for your ORF and the promoter for your marker, you want those to be different promoters, because if they're the same, you increase the chances of recombination and potentially losing all of the genetic material in between those two promoters. So for ubiquitous promoters, we have quite a few different options. It's a little more straightforward if we're being a little more selective in the location or the timing of expression. So to control where you are actually expressing your transgene, it's pretty straightforward. You can use a tissue-specific promoter. There are other options, including conditional expression experiments where you use CRE or Gal4-UAS to control where this gene is expressed. However, these are typically a bit more difficult if you don't already have access to a CRE or Gal4 line. So if you don't already have access to one of those lines, a tissue-specific promoter is going to be the better option. Now, I do want to note that we can take advantage of the tropism of different viruses to enhance targeting. I mentioned before that different AAV serotypes can target different types of cells, or you can pseudotype different viruses like VSV and lentivirus to enhance that specificity. And then to control timing, we just need an inducible expression system, like we mentioned in our example a little while ago. The TET inducible system tends to be the most popular, and this is going to use some combination of activators and or repressors. Now, this can require a little bit of optimization, especially when we're looking at something like lentivirus. For these experiments over here, we transduced cells with expression of this TET inducible system using only an activator here and only an activator here. What we're looking for is no expression until we actually add this activator right here, this doxycycline. But as you can see, we do have this sort of leaky expression that's present when it should not be there. However, if we use both a repressor and an activator, then we greatly reduce that leakiness and we have almost no expression until we add that doxycycline, because the doxycycline will remove this repressor and also kind of facilitate this activator as well, so that you only get this expression when you want. So you can use different combinations of these activators and repressors to get really tight control over the timing of expression, which can be useful, for instance, in those CRISPR experiments that we were talking about earlier. So once we have our promoter all set and we know where and when we want this thing expressed, we have to consider actually what we are expressing, right? So we have a couple of different options. One very popular option is expressing an RNA, for instance, an shRNA or a gRNA for some sort of regulation or potentially genome editing. Let's first talk about some shRNAs, which can be used for knockdown experiments and are very easy to use because we can use databases to just input our gene of interest and get back loads and loads of different options of these 21 nucleotide hairpin structure shRNAs just kind of ready to pop into our vector. Now, these different shRNA options are going to be scored for things like specificity and secondary structures, but those scores are theoretical. You're going to need to test those out to make sure that you validate them. And additionally, it's recommended that you try out at least 3 shRNAs because this is going to ensure that you have that adequate validation. And additionally, it helps to kind of discern which phenotypes are actually real versus those that are potentially off target. Now, another option for RNAs that you can express are gRNAs for CRISPR. So that gRNA is going to be the sort of targeting sequence that helps Cas endonuclease to locate the correct region of your genome and perform cuts or do other modifications depending on whether you're doing knockout, knock in, knock down, or knock up. We can do all of these things with CRISPR. So we can also use databases to search for a gRNA sequence just by inputting our gene of interest, and we'll get these 20 nucleotide gRNAs that are output with a related score. And here that score is going to, again, be highly related to the specificity of that gRNA, but also it's going to balance the sort of on-target efficiency and potential off-target effects. And one thing that I wanted to mention about gRNAs that we have to think about that we don't necessarily have to think about with shRNAs is accessibility. We need to be sure that our target site can be reached by that gRNA Cas complex, and we also have to ensure that a PAM sequence is nearby. Now, if you're interested in learning more about CRISPR approaches, we do actually have another webinar that's happening next month. So be on the lookout for that to learn more about CRISPR approaches. Now, alternatively, our kind of thing to be expressed can just be a standard ORF. If you're wanting to express a protein, there are just a couple of considerations that I really want to highlight here. If you're taking a transgene and you're expressing a gene from one organism in another, it's very worth your time to take a minute to make sure it's codon optimized. This is going to ensure that the sequence that you have corresponds with the most highly used codons in your host organism. This is going to make sure that this translation is the most efficient. Additionally, if you are expressing multiple genes, then we recommend kind of placing your most important gene first, and then the second gene, for instance, if that's your marker, that should go second. This is especially true in lentivirus, as we can see here, where the gene that's kind of placed upstream tends to have this higher level of expression. So if we have our kind of ORF, we have our promoter driving our ORF or our RNA, then our next thing that we have to consider is just what's downstream of that. If it's nothing, great. We just have our little polyA signal and we're good to go. But if you want to express multiple genes, there are different ways that you can kind of situate them beside each other. You can encode them as completely separate cassettes. For instance, if you want to have them have different promoters, you can have them have separate polyA tails. They can be treated completely separately. However, it's worth noting that this is not suitable for lentivirus because if you are making a lentivirus transfer vector, you cannot have an internal polyA signal because that will greatly reduce the titer of your virus. Alternatively, you can have your 2 ORFs that are linked. This is where a single mRNA is produced, but you still can produce 2 separate proteins. The 2 popular linkers that you can use are the IRES, which is an internal ribosomal entry site, where you have just the start of a new translation event in that mRNA. That's a good option, especially if your second ORF is something like a marker. Because the downside here is that your second ORF is going to have lower translation efficiency. Now, one thing to note when using an IRES is that because you have 2 separate translation events, you are going to need a stop codon at the end of that first ORF. Alternatively, you can have a 2A linker, for instance, T2A or P2A. These are kind of skipping linkers or read-through linkers. where a ribosome is going to skip along that mRNA that's going to result in cleavage. Now, for coding this, you are not going to include that stop codon since the same ribosome is kind of going along the entire mRNA. However, the downside here is that you can have additional amino acid residues that are on either side of that cleavage point. So this is kind of to be avoided if those amino acids might interfere with your protein function. And finally, you can do a fusion of those two proteins. So this is where you create a single protein with functions of both of those genes. Now, whichever kind of aspect of these you use, it can sometimes require, again, a little bit of optimization. As we can see over here in lentivirus, if we want to use EGFP with an antibiotic selection marker, how it's oriented is very kind of influential on the expression of that EGFP. So for instance, with puromycin, you have to use this 2A linker. You cannot use a fusion protein. But the opposite is true with neomycin. The fusion protein is actually more efficient than when we use this linker. So these things are actually kind of already taken into account for things like vector builders, design, which we will talk about shortly. But these are things that we need to keep in mind when we're looking at the design of these elements. So finally, I do want to mention a couple of options for markers. One thing, if you've kind of explored the various different both antibiotic and fluorescent markers that are out there, there are a huge variety of different options. At VectorBuilder, for single and dual color, we kind of recommend just gold standard green and red. Things like EGFP and mCherry are very, very widely used, but we are always kind of keeping an eye on new players on the scene, and we do have loads of different options available as well that we're always kind of testing out and seeing how they compare. Now, for tricolor, we recommend kind of looping in this tag BFP or CYPET, but one thing that you have to keep in mind for any and all of these is that you note the excitation wavelength for your fluorescent marker, and you make sure that your equipment, your microscope and your filter are going to complement those excitation wavelengths. This is going to be really important for not only making sure that you can see your fluorescent marker, but also making sure that you don't have interference between your 2 or 3 potentially different fluorescent markers. Now, for either fluorescent markers or for drug selection markers, you can not only use them to select for your cells that have taken up that vector, but you can also use them to enrich for cells with high GOI expression, as we can see right here. So you can use these in a number of different ways, and we're always happy to help with kind of designing those various different elements. So now it seems like a good time to dive into how all of these design considerations and tips can all be taken together in a way that really streamlines your gene delivery experiments. So VectorBuilder is a hub for all things gene delivery, really starting at the very beginning with conception and design. At VectorBuilder, we house over 100 different vector systems, over 1,000 different backbones with basically different considerations and aspects of those vector systems. And we have over 1.2 million different vector components that are in our databases. So if you're looking at a vector like this that can feel a little bit overwhelming, our Vector Design Studio makes vector design really straightforward and really fast. In our Vector Design Studio, you can start with basically just considering your model as well as your approach. So if you want to perform gene overexpression and you want to use a regular plasmid, we have a backbone all set up for that. Same if you want to use lentivirus, we have everything all set where the backbone components are all already in place. And we have the same thing for a variety of different approaches from overexpression to shRNA and CRISPR and CAR expression. And we also have all of these different vector options. Now, once you enter the Vector Design Studio, designing your vector exactly the way you want it can be done in just a few clicks. So you can add your promoter, your ORF, and your marker if you want a marker at all. And if you have a sequence already, you can easily paste it in, or you can search from our database of over 1 million different components. And either way, after you add those components in, you can fully edit, you can add tags to your ORF, you have a lot of different capabilities. And then at the end of this, you can click your Finish Design button and get all of the vector information that you need for free. And of course, we will give you immediate and very transparent pricing for not only the cloning of your vector, but also any downstream services that you might need. So the Vector Design Studio is a great option if you just want a kind of quick and easy design experience, or if you have a kind of more simple vector. Alternatively, if you want more control, we have recently launched our DNA software, VectorBuilder, that is also 100% free. Now, this allows you to have unlimited control not only over your different components, but also with your backbone. So you can visualize, you can edit, you can annotate, you can do anything you want to your vector. But additionally, it has integrated really seamlessly with VectorBuilder. So if you find a little part in your vector that you want to insert a component, let's say you want to insert another ORF, you can still search the VectorBuilder databases to insert those components in just a few clicks. It's super easy. And finally, you can order those vectors that you have designed in VectorBuilder by just quickly sending them straight on over to the VectorBuilder design team and quickly find out exactly how easily and how quickly you can receive those vectors, or if you want them packaged into lentivirus, if you want them made into a library, any and all of those options are possible and easy. So VectorBuilder is really all about gene delivery, but we don't really stop there. We love to see everyone designing vectors on the VectorBuilder website. We actually just recently surpassed 1 million vectors designed on our page. But additionally, we don't just kind of let you go with the design and just kind of say, good luck. We offer very, very extensive free design guidance. And we're realizing how important that design guidance is, because we've recently put out a paper that found that for the vectors that we received from various different labs and industry partners, we found that if they wanted downstream services, when we performed some basic QC on those vectors, we found that more than half of them contained either design or sequence errors. So this design guidance is really more important than ever, and we really want to make sure that everyone knows that this is available to anyone and everyone to make sure that your vectors can perform exactly how you want them. So in addition to this free design guidance and our easy vector design tools, we offer cloning as well as downstream services like plasmid preparation and virus packaging, and even further kind of larger scale experiments, including library screening, stable cell line generation, CRISPR and RNA solutions, and more. Really anything related to gene delivery, that's what we're all about. And we love to talk gene delivery. We are initiating a gene delivery seminar series that starts next week and will occur monthly. Our first speaker is going to be Dr. Priyanka Narayan, who's going to talk about how her lab uses gene delivery to model neurodegenerative diseases. I have a QR code here if you want to register for that, if you're free on September 12th at 1 p.m. But of course, this will be available on our website and on our YouTube channel as well. And in both of those places, we also have extensive resources, including various different webinars for things like IVT RNA solutions, as well as VectorBuilder. And we want to make sure that you have really all of the resources that you need. So we have provided a few different handouts for things like our VectorBuilder software, as well as a virus comparison flyer, and kind of a comparison of doing things in the lab yourself in terms of vector cloning versus having VectorBuilder take care of it for you. So I want to thank you so much for your time, and I would love to continue the discussion and take any questions that you have.